Hello viewers, welcome to this particular lecture on microbiology. In this particular lecture, I shall be discussing some aspects of natural barrier. Natural barrier in human body which protect us from different infection. These natural barriers will include the following structures. They include the skin and the different glands associated with skin. They include the mucous membrane and the lymphoid tissue associated with mucous membrane. They will include different glands like tonsil, the Kupffer cell, the Brunner's gland or the Langerhans cell, etc. They will also include the non-specific immunoresponse, which include the phagocytic cells like neutrophils, like the macrophages, and of course their products. Similarly, it will also include the specific immune response brought about by the lymphocytes and the antibodies. So to start with, we will discuss the skin and the different layers of skin. Now skin is a natural barrier. Skin is the largest organ of human body because it is covering the entire human body. And this particular skin is having two basic layers. One is the epidermis, other one is the dermis. The epidermis again is a relatively thinner layer, but actually it is formed of 5 different layers. And from outside to inside, these layers are the stratum corneum, which is also called the horny layer, and it is the outermost layer, cornified layer, because it is formed of corneocytes. And naturally, these particular cells lost or sloughed off due to loss of environmental moisture or loss of moisture in the skin, and the new cells are formed. In the same way, there is the second layer, which is also called the stratum lucidum. Stratum lucidum is a relatively thin layer. It is also formed of dead cell, and these particular layers are formed of flattened cells called keratinocytes because they contain high amount of keratin and one of its derivative protein called elidin. Coming to the third layer, this third layer is often called the stratum granulosum. The stratum granulosum is so called because they have the granular cells, and these granular cells are actually having some granules. These are the granules, and these granules are called keratohyalin granules, and they are formed of protein which is mostly histidine and cysteine rich. And what is their function? They will hold the keratin filaments together. The next layer is the stratum spinosum. The stratum spinosum is actually the layer between the stratum basal and the stratum granulosum, and they are having microfilaments between the desmosome, and they are also composed of polyhedral keratinocytes. Now coming to the last layer of the epidermis, which is the lowermost layer, this layer is called the stratum basal or the stratum germinativum. This layer is formed of keratinocytes and also melanocytes that will produce melanin, and they are having the Langerhans cell for defense, the Merkel disc for touch reception, etc. And usually it is from this particular layer that the cells of the skin are progressing upward to stratum corneum. Okay.
So how long does it take to reach from stratum spinosum to basal?
Yeah, actually it is not from stratum spinosum to basal,
one.
but the stratum basal to outermost layer, that is stratum corneum. Approximately it takes 14 days. Okay. Now coming to the next layer, which is the dermis. Now the dermis is very much living layer because it has the blood vessels, it has the lymphatics, and broadly it is having two different layers. The upper layer is called the papillary dermis and the lower layer is called the reticular dermis, which is more or less in continuity with the connective tissue below. But actually speaking, this dermis, you can think it is such a thick layer. It is formed of different tissue, and they include the muscles, they include the sebaceous gland, they include the hair follicle, they include the nerves, and of course they include the sweat glands. So all these particular structures are there, and just below the dermis there will be the adipose tissue layer which will support the dermis and also will keep the dermis well nourished. So this particular skin actually has got some function. It will prevent the entry of pathogen directly as a mechanical barrier, and also it will lubricate the skin with different glands. One such gland is the Langerhans cell. What are Langerhans cell? They are actually specific dendritic cell. Why dendritic cell? Because they have projections. And these dendritic cells are actually antigen presenting cells. They present the antigen to T lymphocytes. And they have the small Birbeck cell, which is actually small granules and mostly is present in the stratum spinosum. Now what do they do? They are actually, this is the location of the Langerhans cell which gets activated. They get activated due to natural stimulus, due to UV ray, different things are there, and they gradually, as they get activated, they get inward into the dermal layer, and from there they present the antigen into specific MHC 1 or 2 and ultimately binds with T cell. So in one way they are getting activated, they are trapping the particular bacterial antigen and transferring it to T cell for destruction. So this is the function of Langerhans cell in the skin. Now there are glands called sweat glands. Sweat glands as such, they are mostly present in the dermis, but their opening is in the outermost layer of epidermis, that is the stratum corneum. And these particular glands are of two types. The common type is the eccrine gland, which is present all over the body producing maximum sweat, and the other type of gland is the apocrine gland, and they are produced mostly from certain areas like the armpit. They are producing more secretion and they have the purpose of cleaning the skin, of controlling the body temperature, and also trapping bacteria. Apart from that, you must also understand that these apocrine gland actually they are lost partially and they are again resynthesized. But in case of eccrine gland, more or less the structure is maintained, as you can see in this particular slide. Now coming to the next important gland, which is the sebaceous gland. Why it is so called? Because it produces sebum. And this sebum actually is lubricating the hair because they are present along with the hair follicle. So these sebaceous glands are follicular cells and they produce sebum. And what are their functions? Sebum actually, these are the sebaceous gland actually under the microscope, and it will protect against germ, it will act as a barrier. Similarly, it delivers different antioxidant or antimicrobial lipid or even hormone like pheromone. Similarly, it will contain some different fatty acids which will give the characteristic odor, and they also produce vitamin E to the upper layers of the skin. Similarly, they have important thermoregulatory function, and of course they can actually make the skin waterproof. So there are different functions, and of course the most important function in this context is it will protect from bacterial infection to some extent. Now coming to the next important part or next important protection given by the mucous layer. So the mucous layer mainly containing different immunoglobulins like IgG and IgA. They also produce lysozyme, and this mucus is actually associated with lymphoid tissue. When it is present in the gut, it is called GALT, meaning gut associated lymphoid tissue. When it is present along with the respiratory tract, it is often called BALT, that is bronchus associated lymphoid tissue. But basically the most important component of mucus is mucine, and this is the branched mucine having disulfide linkage and binding with different molecules like lipid, like carbohydrate, like calcium ion. Different things are there, but broadly mucine are of two types. They are the sulfur containing mucine, they are called sulfomucine. They are also the sialic acid containing mucine or called the sialomucine. And what do? Primarily they will bind with different molecule, will ultimately prevent the entry of bacteria into the inner tissue. This is one. And the second is it is also controlling calcium hemostasis between the muscles and the connective tissue. Now let us consider some aspect of the respiratory tract. Actually in the bronchus, these are the mucous gland, and this mucous gland will produce a secretion of mucus or mucine, which will actually produce the lining of the trachea, and this lining will be active. There will be some ciliated cells. So whenever there is a pathogen entering, these ciliated cells will put them out. This is one. Or they will act as a filter, preventing the entry of the pathogen into the lung. So that way they are protecting. And this particular mucus production should be clean. If the mucus is actually too much, then we suffer from this acute cold or cough because it is thickened and it is blocking the respiratory tract. Now let us come to another type of cell which is also associated with the respiratory tract and with the gut, and broadly they are called the M cell or the microfold cell. What are they? Actually, microfold cells are specific cell in the endothelium or in the epithelial layer. This microfold cell will allow the entry of bacterial antigen, and as it enters into the inner tissue, it will bind with specific cells which are similar to macrophage, and this macrophage or reticulocytes will ultimately take the infection to MHC class 1 or class 2, and ultimately they will be destroyed by T cells. So in one way, M cells or microfold cells are trapping bacteria, trapping antigen, activating the lymphatic tissue, and most importantly, it will make sometimes the bacteria immobile. Say for example, if it is present in the gut and it makes the particular bacteria E. coli immobile, then at that particular point it will produce vitamin D. So these are the functions of the M cell. Now let us discuss some aspects of the alimentary canal and the glands associated with the alimentary canal, which act as a natural barrier. Now the gatekeeper of human body is the tonsil. The tonsil is divided into anterior tonsil, palatine tonsil and posterior tonsil, and of course it is having the lingual tonsil. So basically only a part of the tonsil, which is the palatine tonsil, that we can see, but the upper part, which is actually getting above the tonsil as anterior tonsil or adenoid tissue, we don't see them because that is behind the nasal cavity, and the lower one below the tongue, that also we don't see. But what is the importance? This particular tonsil produces the lymphocytes, which are actually activating T cells, and they are adhering to bacteria or bacterial pathogen. So thereby a tonsil is a very important structure in human body which prevents the entry of bacteria into the alimentary canal. But coming to the next important line of defense is actually a digestive gland, and this digestive gland is the parotid gland, the submandibular gland and the sublingual gland. Actually the largest gland is the parotid gland, and the submandibular gland is the middle one, and the smallest one is the sublingual gland. The parotid gland actually produces the saliva, but the maximum saliva up to 70% comes from submandibular, and maybe parotid producing another 20-25, but only 5% comes from the sublingual gland. And this saliva actually contains a very important enzyme which is lysozyme. Apart from lysozyme, they also produce mucine, so they are both bacteriostatic. At the same time, they can kill bacteria by the action of lysozyme. So this lysozyme is ultimately preventing bacterial growth inside the mouth cavity, and at the same time the bacteria in the food which has entered the mouth cavity will be killed.
Sir, we also know that bacteriophage can produce lysozyme. So is there any difference between bacterial lysozyme and human lysozyme?
Yes, there is a difference. Actually, bacteriophage lysozyme is more versatile. It can kill the particular bacteria at multiple sites, whereas human lysozyme has got certain limitation in comparison to bacteriophage lysozyme. Okay? Now, apart from this particular lysozyme, saliva has got other function. What is that? It produces immunoglobulin like IgA. It produces different factors that control microorganism growth and thereby regulate the number of microorganisms. It also is protective because it prevents the formation of dental plaque, which is actually formed by the bacterial film, biofilm. So this is also prevented. And most importantly, it is also preventing ascending infection in the parotid gland. So that is also prevented. So saliva is a very important protective fluid which is actually present all along the lining of the mouth cavity. Now coming to the next important cells, which are single cell gland called the goblet cell. And this goblet cell also produces mucus or mucine, and they are coming out through this microvilli, and these secretory vesicles continually will maintain the lining of the digestive tract. And these are the actual size of the goblet cell present along the lining of the intestine. But the most important enzyme gland is the gastric gland, present along the lining of the stomach. And this gastric gland, one of the cells is the parietal cell and the oxyntic cell, which is producing the HCl. And this HCl actually is formed by the selective entry or exit of hydrogen ion into the lumen, and also the chloride ion diffused into the canaliculi. So H and Cl will combine and HCl will be produced, and this will reduce the pH almost to 2 to 3 in the stomach lumen. And as it is coming down, it will kill majority of bacteria, though some acidophiles will be surviving at this low pH. So this particular HCl production is the second line of defense in human alimentary canal. Okay. Now coming to the next important gland, which is the Brunner's gland. Brunner's gland is present all along the alimentary canal, and it produces mucus or mucine, protecting the alimentary canal or the inner layer of the alimentary canal. Apart from protection, they make the alkaline condition, they will lubricate the intestinal wall, or they can actually produce some hormone helping in digestion. But mostly it is protecting. Coming to another very important cell, which are the macrophage or bacteria trapping cell present in liver, which are called the Kupffer cell. Actually, the Kupffer cell is producing different interleukins or molecules which are defense associated to the liver, and this particular interleukins will prevent the growth of different pathogenic organisms. So Kupffer cell in one way is cleaning or devouring the debris, and at the same time Kupffer cell is also helping in arresting the growth of pathogenic organism. But let us consider a particular organ which is the neural tissue or the brain or the spinal cord. There are some specific endothelial cells which are called microglia. And what are they doing? These microglia are actually phagocytic cells. They will phagocytosize on different bacterial organism on one side, and at the same time they may be active to produce different defense related molecules, and these defense related molecules may be interleukin, and they will activate the defense system. So if there is any infection in the nerves, this is treated by the microglia. Let us consider some aspects of the urinary system. In the urinary system, the most important structure is the urinary tubule inside the nephron. They produce a typical mucoprotein called tam horsfall mucoprotein present within the urinary tubule. They actually trap the bacteria and make them immobilized. They don't allow them to move, and thereby the bacteria may be arrested, the growth may be arrested, and the urinary tract infection may be arrested. Now this gentleman, that is Mechnikov, in 1882 discovered a very important cell, and that is called macrophage. So when I am talking about macrophage, you should know these are a systemic, general system of blood vascular system of alimentary system. They will all have this particular macrophage. And macrophage directly, they are the phagocytes which will devour on bacteria, as you can see in this particular electron micrograph. And they have the unique ability to metabolize a particular amino acid called arginine. And this metabolism will produce either nitric oxide, which is a killer macrophage. Another one is ornithine, which is a repair macrophage. So accordingly, the killer macrophage are called M1 macrophage, and the repairing macrophage are called the M2 macrophage. But the basic function of macrophage is to trap a bacteria, take it inside the vesicle, and thereby with the help of lysozyme they will be killed. So these are the different stages of bacterial trapping and killing within the macrophage. Coming to another very important cell which is present in the blood vascular system, which is the eosinophil, having a trilobe nucleus. This eosinophil may actually produce some anti-allergic protein like histamine. It may produce some reactive oxygen species like superoxides, which is of course not good. It may also produce a series of cytokines like IL-1, IL-2, IL-4, IL-5, IL-6, IL-8 and IL-13. And these interleukins are defense related. They can also fight the viral infection, and at the same time they may induce allergic response which will ultimately check the bacterial growth. So basically eosinophil is producing protein which will prevent pathogenic growth within human body in different organs. Another very important cell which is of course present in the blood is the lymphocytes, and these lymphocytes are actually trapping the bacteria, and they are of two types. They are the T cells and the B cells, and these B cells directly will be activated by the T helper cells, and ultimately they will produce the antibodies the moment it enters into human body, that is the humoral immunity. Whereas the T helper cell will activate macrophage for phagocytosis, and at the same time the CD4 directly can produce the killer T cell which will destroy the infected tissue. So the T cell and the B cell basically will be active in the presence of some foreign antigen, and they are also non-specific in nature. And this brings me this type of immune response which is called non-specific. What is non-specific? Non-specific means that particular immune reaction is not specific, like the one which is brought about by different interleukins, the one which is brought about by interferon, and this particular non-specific infection will ultimately bring about the change in physiology like fever, but ultimately it will restrict the growth of pathogen, not a specific type, any type. And these are certain non-specific immune reaction brought about by interferon, brought about by interleukin, which will activate macrophage, which will activate the natural killer cell, which will activate the T cell, which will allow the production of specific lymphocyte, but ultimately to check the growth of pathogenic organism within human blood vascular system. The last one, which is the specific immune response, is more or less specific to a particular pathogen, and that will make the pathogen immobilized, and at the same time they will kill it by phagocytosis. That is brought about by a particular molecule called opsonin. This C3b or opsonin is actually deposited on the bacterial wall, and they are specifically taken to some phagocyte, and they are engulfed by the phagocyte and they are killed. At the same time they may be activating specific membrane attack complex, and that will also lyse the cell or the bacterial cell into pieces, lysis of the bacteria. So in this way the specific immune response will ultimately make some specific bacteria killed or will protect us from specific infection, and this is always pathogen specific.